Hey everyone, course review day 123 from beautiful Torrey Pines. You all know it. Right, csRNA-seq, what we basically did is day one, we took our total RNA and purified our small RNA. We eluted that and then precipitated overnight. So now that we have our small RNA, we separate it into the untreated one, which is our input. And today we did a 5' enrichment. What does that actually mean? We have different modifications like the cap, triphosphorylation, diphosphorylation or monophosphorylation. This is especially the microRNAs and other small RNAs. And this is basically our short initiated RNAs, which is sort of the TSS RNAs, right? And this is what we want. And so we depleted this using two ways, which is the terminator and the CYP. CYP basically terminator removes only these ones here, right? While CYP removes all the different phosphates. And as we are hanging out here, these samples are precipitating. So as we're hanging out here, our samples are precipitating, and tomorrow, at least after we spin down, we will have our capped enrichment and we have our input. So this has basically all the different phosphorylations and cap, and this has our cap. And the first thing we're going to do to all samples is we use this pyrophosphatase called RPPH, which make everything monophosphorylated, whether cap, triphosphate or diphosphate. And of course, what's monophosphate will just say monophosphate. We do this because you actually need 5' monophosphate for the adapter ligation on the 5' end. The most tricky thing tomorrow about this will be that we're going to add a ton of PAC in here. PAC is a crowder, and the reason we add it is so we increase the PAC concentration during the 3' adapter ligation, thereby increasing its efficiency. So this will take us an hour, and then on top of that we add the next master mix, which is basically the T4RNA ligase 2, and it's the truncated version of that. So now that we have the 5' phosphate, we're gonna ligate on the adapter. So the reaction that is catalyzed here, and this will be sort of the green tube tomorrow, is the 3' adapter ligation, which takes the 3' OH with the adenylated adapter to add the 3' adapter. Now, to avoid adapter dimers, because we still need to ligate on the 5' adapter, the next step we're going to do is we anneal a primer. And why do we anneal a primer? It's because T4RNA ligase 1, which we use for the adapter ligation, likes single-stranded nucleic acids, but not double-stranded. And by making it double-stranded, we basically block all the adapter, the bound one, but also the unbound one from adapter ligation. Once this is annealed, now as the next step, we can add on the 5' adapter. And this is done by T4RNA ligase 1. Out of all the steps tomorrow, I forgot to say, this is the rate-limiting step and the expensive one. Well, this will take us another hour. This is the yellow tube. Once this adapter is on, we can just add the RT master mix, and the reverse transcriptase will just sort of prime off, and using the annealed primer will cause cDNA. This will then have our 5' adapter and our 3' adapter here that we can then use for amplification using PCR. And in this PCR reaction, we will also adjust the barcode for our unique identifiers. So for tomorrow, we're most likely going to stop with the RT. The RT can be done for 1 to 2 hours and then just kept on ice overnight. Well, if we're very ambitious, we might even do the PCR. At the last day then, we'll just need to do the PCR if we haven't done it, purify it, run it on a gel, cut it out, and then Thursday to Friday, we take the gel cut out, elute it again overnight, Or if you're very motivated, we can elute it within 20 minutes and load it on the sequencer. So this is a quick course overview. And again, tomorrow is usually the busiest day of all. So I thought just keep repeating that we basically have cap and input. And for input, we just use 50% of our master mixes. Master mix 1 is decapping. Master mix 2 is adding the 3' adapter. Then we use the PCR machine to anneal the primer. Master mix 3 is to add on the 5' adapter. Master mix 5 is to reverse transcription. And master mix 6 is to make the PCR. And so with understanding that tomorrow will not be too stressful. With that, enjoy the sunset. I'll see you tomorrow.